Chapter 8, Cellular Reproduction. Biology and Society, Why Cellular Reproduction Matters. About 9 months before you were born, you were just one single cell. And from that microscopic beginning, every cell in your body was created through cellular reproduction. Even today, millions of cells are reproducing themselves in your body every second, and errors in the process can be deadly. A female zebra shark in Australia laid eggs and reproduced a daughter without the involvement of a male. And this process is known as parthenogenesis. So all of the genes for the daughter shark came solely from her mother. Parthenogenesis is very rare among vertebrates, but it has been seen in about 70 species. So what is accomplished by cell reproduction? We know that reproduction causes the birth of new organisms. So if it is cell reproduction, that means it is the birth of new cells. So one parent cell divides or splits or reproduces into two daughter cells. And in order to do that, the parent cell has to first double or duplicate its chromosomes, that is the genetic material which contains DNA. It is made up of DNA and protein. So first the genetic material is duplicated, and then the other stuff, which is not the chromosomes, the cytoplasm and all, it is split equally into the two daughter cells. So why do cells reproduce? Cells reproduce for the following three reasons. Number first, damaged and lost cells have to be replaced. You know, we are shedding our skin cells every day, and those have to be regenerated or replenished, all the cells that we've lost. So we are constantly forming new skin cells in the deep layers of the skin, and those cells are moving outwards. And the outermost layers of cells, we are shedding them off. So it is replacing of damaged or lost cells. It also permits growth. So imagine a child growing up into an adult. There's an increase in the height. How do you think it is happening? The bones are increasing in length. The muscles are increasing in size. It's because of cell reproduction. And in general, it allows for reproduction, for production of offsprings. These are the different functions of mitosis. Reproduction can be two types, asexual and sexual. So let's take asexual reproduction first. In this type of reproduction, unicellular organisms, that means organisms that have only one cell in their body, they usually reproduce by just splitting into two. So the two offsprings are exactly identical to their parent cells. They are genetic replicas of their parent cells. So these resulting offsprings can also be known as clones, okay, because these are a group of genetically identical individuals. So mitosis is that type of cell division which leads to asexual reproduction. So whenever a certain part of our body, for example, there's a bone fracture and the fracture has to be repaired, that means new bone cells have to be formed. So this is where mitosis occurs. So growth and maintenance of multicellular organisms occurs by means of mitosis or asexual reproduction. The second type of reproduction is sexual reproduction, and it involves the production of gametes or sex cells. So in case of human beings, there is the females make the egg or the ovum, and the males will make the sperm or the spermatozoa. And there has to be a union of the two called fertilization, and it leads to the production of a new offspring. And the type of cell division by which the sperm and the eggs are formed is known as meiosis. So meiosis is very different from mitosis. So that means for sexual and asexual reproduction, we have these two types of cell divisions: meiosis for production of offsprings and mitosis for growth and maintenance. Now let's take a eukaryotic cells. Where would you find the genes? Most of the genes would be located in the chromosomes that are present inside the nucleus. There are some genes that are also present in the genetic material that's present inside the mitochondria or chloroplasts in case of plant cells. So each eukaryotic organism, think of it as a long thread-like structure, and it contains DNA, and then there are some proteins on it. And this chromosome contains genes which are linearly arranged. So how many chromosomes would a particular eukaryotic organism have? It depends on the species. Chromosomes are made up of long thread-like structures, and those long thread-like structures in turn are made up of DNA and protein, and these long thread-like structures are called chromatin. So the proteins that are present, they help organize the chromatin and also control the activity of the genes. So most of the times when the cell is not dividing, you would find that the genetic material of the cell is in the form of chromatin. It is only at the time of cell division, just before the cell is about to divide, that the chromatin, it shortens and becomes thicker, so it condenses, and then it is known as a chromosome. So how are the DNA and proteins packaged in the chromosome? It is a particular structure. It's known as the beads on a string structure. The beads are the protein molecules. They are the histones, special type of proteins. And the string is DNA. And nucleosome, this particular, the way the DNA winds around the protein is known as a nucleosome. It consists of the DNA thread-like structure, which is wound around several histone molecules. You see here the structure of the nucleosome, the beads on a string structure, and it all starts from the DNA double helix and how it winds around the histone molecules, those purple colored beads, and then it becomes more and more compactly arranged, and then it all comes into the X-shaped chromosome structure at the bottom of the slide. The central constriction is known as the centromere, and the two arms of these X are known as the sister chromatids.
Several levels of DNA packing make up the compact chromosomes seen in a metaphase cell. Even when a cell is not dividing, some of the DNA is folded tightly. The degree of DNA packing makes certain genes available or unavailable for transcription. Chromosomes consist of loops of supercoils, which are made up of tight helical fibers. These fibers are composed of nucleosomes, which consist of DNA wrapped around protein molecules. In the fiber, the DNA is unavailable for transcription. Each nucleosome is a cluster of proteins called histones. Nucleosomes change shape and position to allow RNA polymerases to move along the DNA and read the genes. Chemical modification of DNA and histones may affect chromatin structure and access to specific genes.
Now, before a cell undergoes cell division, the DNA molecule or the genetic material has to be duplicated. Means it has to make 2 copies of it in order for each daughter cell to receive 1 copy. And then you can see the long thread-like chromatin getting shorter and thicker into a chromosome, and the chromosome looks like that X structure with the 2 sister chromatids. These are joined at the centromere. The main event of cell division is the separation of the 2 sister chromatids. So the sister chromatids of each chromosome, they are separated, they get split into 2, and then each sister chromatid would mature into a full-fledged chromosome. This figure illustrates in very simple manner how a single chromosome, you see just before the cell division, it would duplicate, so it becomes X-shaped and has these 2 sister chromatids. And during cell division, these 2 sister chromatids would be split, would be separated, and then they become, their own full-fledged chromosomes. A cell cycle is the life cycle of a cell from its own birth to the time that it reproduces and divides or splits into 2 daughter cells. So it happens in a cyclical or a circular fashion, and a cell ends up spending most of its lifetime in a stage known as the interphase. So that's 90% of the cell cycle. And this is when the cell is actually preparing to go into cell division. The cell is growing, the cell is maintaining, and then the cell is doubling up everything, doubling its DNA, doubling its protein, just getting ready for the cell to divide. You see the eukaryotic cell cycle in this figure. All of the interphase accounts for 90%, and then just the rest, the 10% is when the cell actually divides, that is the mitotic phase. The interphase can be further subdivided into 3 subphases. The first one is the G1 phase. G1 stands for first gap. The second is S phase, which is synthesis of DNA, and the G2 is the second gap. Although you see this term gap doesn't mean that the cell is resting. The cell is actually very, very active, and the cell is actively preparing to go into cell division. The interphase is followed by the mitotic or the M phase. And in mitotic phase, there are 2 things that happen. Mitosis, which is division of the nucleus and its contents into two daughter cells. And then this is followed by the cytokinesis, which is division of the cytoplasm and all of its organelles into 2. So it is this combination of mitosis and cytokinesis that ultimately gives rise to 2 genetically identical daughter cells. So mitosis is further subdivided into these phases or subphases: prophase, metaphase, anaphase, and telophase. Now we're going to learn about the entire process of mitosis in this figure. It starts with interphase where the cell is preparing, then it goes into prophase, so I, P, then it reaches metaphase, M, anaphase, A, telophase, it, mat. These are the different subphases of mitosis. In interphase, the cell is simply preparing for cell division, so there are three subphases. G1 where the organelles get duplicated, S phase where the DNA is doubled, so it's DNA replication, and the third one is G2 where the protein gets doubled. So this is where the cell is preparing. You see the long thin chromatin inside the nucleus, you can see the nuclear membrane, and you see these two organelles called centrioles, which will play a very important role in cell division. Then we move to prophase where the cell has just entered cell division. The centrioles, they start moving apart. Okay, they are moving in opposite directions. They want to move to the two opposite sides or opposite poles of the cell. They start forming the mitotic spindle. The nuclear membrane starts getting fragmented. Okay, the chromatin has become shorter and thicker, and it is assuming the shape of a chromosome with the sister chromatids. By late prophase, the two centrioles have reached the opposite poles. They have radiated the spindle microtubules, these thread-like structures, which have gone and attached to each chromosome at the centromere. You can see the chromosomes with their sister chromatids, and the nuclear membrane has totally disintegrated. The next phase is metaphase, where the chromosomes align themselves in the center of the cell, which is known as the metaphase plate or the equatorial plate, which is an imaginary plate about equidistant from the two poles. The centrioles are at the opposite poles, and the spindle microtubules have attached to the centromeres in the center of each chromosome. The next phase is anaphase, where the spindle microtubules shrink, and as they shrink, they pull the centromere, and in doing so, they separate each chromosome into their two sister chromatids. You see, every chromosome has been split, and the sister chromatids on this side, they start moving to, on one side, they start moving to one end of the cell, and the sister chromatids on the other side, they move to the other end. And then finally, in telophase, is exactly the opposite of prophase. Whatever was happening in prophase, you just reverse those. So nuclear membrane starts reappearing, the chromatin material becomes long and thin. It is no longer a chromosome, it is chromatin. You see the centrioles are no longer radiating the microtubules. And as telophase is occurring, simultaneously overlapping it, cytokinesis starts, which is division of cytoplasm, and it starts with the formation of a cleavage furrow in the middle of the cell. Now, the next few slides will show a diagrammatic representation as well as scanning electron micrographs of cell division. So the mitotic spindle plays a very important role because the chromosomes and their movements depend on it. And this spindle is a football-shaped structure which is made up of microtubules, and it is the microtubules that cause the separation of the two sets of daughter chromosomes. Cytokinesis is different in animal and plant cells. In case of animal cells, we see the formation of a cleavage, which is appearance of a cleavage furrow, which is an indentation at the equator of the cell. This figure illustrates cytokinesis in animal cells.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
In plant cells, cytokinesis happens by the formation of a cell plate. And this happens when vesicles that contain materials for cell wall building, they accumulate in the middle of the cell and then they fuse. So this results in the formation of a membranous disc, and this is known as a cell plate. This figure shows cytokinesis cell plate formation in plant cells.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
In this time-lapse video of cell division in a newt lung cell, you can see a process that normally takes more than an hour occur in less than a minute. This newt cell has already started prophase, the first stage of mitosis. Chromosomes have condensed and become clearly visible, contained within the nuclear envelope. In the cytoplasm, the mitotic spindle begins to form during prophase. During prometaphase, the nuclear envelope breaks up. The chromosomes become even shorter and thicker. The microtubules of the mitotic spindle attach to the chromosomes and move them toward the equator of the cell. In metaphase, the chromosomes convene at the metaphase plate, an imaginary plane equidistant from the spindle poles. Anaphase begins when the paired centromeres of each chromosome separate and the sister chromatids, now full-fledged chromosomes, are moved to the poles. The poles of the cell are moved apart, stretching the cell. During telophase, two daughter nuclei form and the chromosomes become less tightly coiled. Cytokinesis begins during telophase, resulting in two identical daughter cells. Credit Conley L. Ryder, New York State Department of Health.
Now, when we talk about cell division, we also need to mention cancer, because cancer means a cell division that has gone out of control. We are all healthy and living. It's because our cell division is very tightly controlled. We have stop and go ahead signals, and how many times cell would divide, when the cell has to die. Everything is very tightly regulated. But when a person gets cancer, this is when the cell cycle control system fails. It could be genetic, it could be environmental influence, or it could be both. So cancer can be called a disease of the cell cycle. Okay, cancer cycles do not respond to normal stop signals of the cell cycle control system. They just keep on dividing uncontrollably, and sometimes may also invade other tissues. So a cancer can start at a particular location but could invade into other tissues. For example, a prostate cancer cell can go into the bone and form a secondary tumor there. So this abnormal behavior starts with a single genetic mutation. It could be just one mutation, and then this leads to a chain of events, and that would cause a lot of cells to become abnormal. So cancer cells can form clumps or masses of abnormally growing cells, which are known as tumors. If the tumor is restricted to a particular location at the original site, then that lump is called a benign tumor. It's not dangerous. However, those tumors where the cancer cells have spread into neighboring tissues or other parts, forming new tumors, and it could have adverse effect on another organ function. Those are known as malignant tumors. So even when a person has a malignant tumor, it's called a cancer. And this process by which the cancer spreads, okay, when a cancer has spread from beyond its original site, that is known as metastasis. This figure here is showing the growth and metastasis of a malignant breast tumor. You see the red one is the tumor, and it is growing inside the breast tissue. It slowly starts spreading, and it invades neighboring tissues, and then finally it metastasizes. That means they spread through lymph and blood vessels to other parts of the body. There are three main types of treatments used for cancer. First one is surgery to remove the tumor. Okay, this is the first step and the most effective one. The second one is radiation therapy, where the tumor cells are exposed to high energy radiation. We have different types of radiation, and these radiation are supposed to harm the cancer cells more than normal cells. It's not that the normal cells do not get harmed. Yes, they do, but the harm is more in cancer cells. And radiation therapy is effective against those malignant tumors that have not yet spread. The third treatment is chemotherapy, which is the use of chemicals or drugs to stop the cell division. And this is used to treat tumors that have spread throughout the body. One of the new methods that is very promising is the use of immunotherapy. In this, experts use the body's own immune system to fight against the cancer. And these immune systems, the components that are used can be either what the patient already has naturally present, or it could be injected into a patient. And many scientists believe that immunotherapy is a major breakthrough, and it is the future of cancer treatment. See, cancer, like most diseases, has two components, genetic and environmental. We cannot control the genes, but we can definitely control our environmental influences. And there are some minor things that we can follow in life. Just having a healthy lifestyle reduces our risk of getting cancer. If we do not smoke, if there is no alcohol, if we are exercising regularly, we avoid overexposure to the sun, we are eating healthy food, high fiber, low fat, we are not eating a lot of junk food or processed food, we perform self-exams, we go to the doctors on a regular basis. These are just things that if we follow, we could definitely reduce our chances of getting cancer. We are done with mitosis or asexual reproduction and moving on to meiosis, which forms the basis for sexual reproduction. Sexual reproduction causes the production of offsprings that contain unique combination of genes from both the parents, and this depends on meiosis followed by fertilization. So before we understand meiosis, let's talk a little bit about human chromosomes. We know that individuals of the same species, they have the same number and types of chromosomes. So in case of human beings, if you take any somatic cell means a cell that is not a sex cell. That means a cell that is not a sperm or an ovum is a somatic cell, and it contains 46 chromosomes. And a geneticist can produce or create a karyotype using this, using all these 46 chromosomes. What the technician can do is break open a human cell when the cell is in the metaphase stage. It could stain the chromosomes using different stains and dyes, take picture of the chromosomes with a microscope, and then arrange the chromosomes in decreasing order of their size. So the largest go at the beginning and the smallest go at the end. And when the chromosomes have been arranged like that, they are said to be arranged in a karyotype. So the figure on the right side, which shows you those 23 pairs, those are chromosomes in a karyotype. So you see our chromosomes, we have pairs of chromosomes. So when we say 46 chromosomes, what we mean is actually we have 23 pairs of chromosomes. So the first one is a pair number 1, the second one is pair number 2, pair number 3, and so on, all the way up to pair number 22, which are all of our autosomes. While pair number 23, this is a male karyotype, so you have an X and a Y chromosome. If it was a female, you would have two X chromosomes. So this is how a karyotype where the chromosomes are arranged looks like. If you just take chromosome pair 1 and then you enlarge it on the left side, this is what you see. So we call these two pairs of the same chromosome as pair of homologous chromosomes. Now, each chromosome, if you take those two threads of each chromosome, are called the sister chromatids. And the constriction, the waist-like constriction in the center, if you remember, is the centromere. So homologous chromosomes are the same pair of chromosomes, and they resemble each other in the size, the length, and the position of the centromere. They also carry genes controlling the same inherited characteristics. For example, the same pair might have genes that control your eye color or your height. So as mentioned before, in our 46 chromosomes, we have 22 pairs of matching chromosomes called autosomes, while the last pair is a pair of sex chromosomes, and these are two different chromosomes, X and Y, which would determine whether a person is male or female. So if in case of female, there would be two X chromosomes, while in case of male, there would be one X and one Y chromosome. So the life cycle of a sexually producing organism like human beings, it starts from birth, and then it goes all the way to when the individual creates or gives birth to an offspring. So this is the human life cycle. The human life cycle is depicted here. If we start from the left side, so you see an adult male and an adult female. These are multicellular diploid adults. Each cell in their body has 46 chromosomes, that means 23 pairs of chromosomes. Inside the ovary of the female and the testes of the males, a special type of cell division which will occur, which is known as meiosis. And by meiosis, this female will make an egg cell and the male will make a sperm cell. And when sexual intercourse occurs, then the sperm cells are introduced into the reproductive tract of the female, and the sperm cell goes and fertilizes the egg cell, that is fertilization. And when fertilization occurs, the egg cell and the sperm cell, which were haploid or had half the number of chromosomes, that means instead of 23 pairs, they just had 23 chromosomes. So when fertilization occurs, that 23 plus 23 gets back to 46. A new life is formed with this first cell. This is the zygote, okay, and it contains 23 chromosomes from the maternal side and 23 from the paternal side. And then this particular diploid zygote divides repeatedly by mitosis. It grows into, it looks like a mass of cells, and that mass of cell becomes an entire new organism. Human beings are called diploid organisms because all of our body cells contain pairs of homologous chromosomes, while only the sperm and the ovum are in the haploid state, because a haploid cell has only one member of each pair of homologous chromosomes. So there's the haploid sperm and the haploid ovum or the egg, which fuse together by a process called fertilization. And this fertilized egg is now called a zygote, and this is again diploid. This figure here very simplistically explains how meiosis halves the chromosome number. If we start on the left side, think of this cell with only two chromosomes, one red and one blue. So the chromosomes will duplicate before the cell division, right? That means you look at the red chromosome, it has become X-shaped, and the blue chromosome has also become X-shaped. So each chromosome now has the sister chromatids, and you see the centromere. Now, if this was mitosis, what would have happened? That the red chromosome would have separated into its two sister chromatids, while the blue one would have also separated into its two sister chromatids. But in case of meiosis, meiosis is divided into meiosis 1 and meiosis 2. So in meiosis 1, instead of the sister chromatids separating, it is simply the separation of the homologous chromosomes. So after meiosis 1, you will see that this particular cell with the red and the blue chromosome has split into two daughter cells. One daughter cell has only red chromosome and the other one has only blue chromosomes. Now in the second step of meiosis, which is meiosis 2, each cell would behave just like it is undergoing mitosis. And now you see the red chromosome splitting into the two sister chromatids and the blue chromosome also separating into the two sister chromatids. So at the end of meiosis, instead of having two cells, from one single mother cell or parent cell, now 4 cells have been produced, and each of these 4 daughter cells have half the number of chromosomes. So meiosis is a very special type of cell division which causes the production of haploid gametes in diploid organisms like human beings. This is similar to mitosis, but there are 2 main differences. The first difference is that the number of chromosomes gets halved at the end of meiosis. And the second difference is that in case of meiosis, there is an exchange of genetic material, okay, pieces of chromosomes between homologous chromosomes. And this exchange is called crossing over. This occurs during the first prophase of meiosis, and this is what causes a combination, permutation combination of genetic material and brings in more genetic diversity. This slide is showing the first stage of meiosis, that is interphase. This slide shows meiosis 1 of the entire meiosis division. So you have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. In case of prophase 1, you see the special event occurring, sites of crossing over where genetic material would be exchanged and brings in new genetic combinations. Then metaphase 1, anaphase 1, in metaphase 1 kind of exactly like what mitosis looks like. In anaphase 1, here instead of the sister chromatids getting pulled apart, it is the pair of homologous chromosomes that's split up, and then telophase 1 and cytokinesis is similar. The next phase of meiosis is meiosis 2, which consists of prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. This is where, remember, we start with 2 cells that have come, just come from meiosis 1. And now each of the cell would behave just like it is undergoing mitosis, and it involves the metaphase alignment on the equatorial plate. In anaphase 2, you see the splitting of the sister chromatids.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So, quick review of mitosis and meiosis. For both of these cell divisions, the chromosomes would duplicate only once. That happens in interphase before the cell goes into either mitosis or meiosis. Mitosis is asexual reproduction, which is used by the body for growth, for repair, all of that. And here, one cell would split into 2, and both the daughter cells would be genetically identical to the parent cell. While meiosis is for sexual reproduction, and this causes a combination of genetic material. Okay, when the sperm fertilizes the ovum, then a new diploid fertilized cell is produced, and the main difference between meiosis and mitosis is when the chromosome number gets halved, and also that in meiosis there is crossing over. So this is a comparative analysis of mitosis and meiosis. Everything that makes meiosis different from mitosis occurs in meiosis 1, right? Crossing over, and the number of chromosomes getting reduced to half. So meiosis 2 is identical to mitosis because it's separating sister chromatids. However, unlike mitosis, meiosis 2 gives rise to daughter cells with half the number or haploid set of chromosomes. Meiosis in sexual reproduction produces genetic variety in the offsprings. In the next slide, we will see how meiosis does this. But what happens is in meiosis 1, during metaphase 1 stage, when chromosomes align in the equatorial or metaphase plate, how the homologous pair of chromosomes would align is a matter of chance, if it's a side by side or how. So for a species that has more than 2 pairs of chromosome, every chromosome pair would orient independently of all others at metaphase 1. This figure shows the results of alternative arrangements of chromosomes at metaphase of meiosis 1. If you see in possibility 1, the 2 blue chromosomes are on the left side, but in possibility 2, there's a blue chromosome on the left and below it is a red chromosome. So this is how there are 2 alternative ways in which chromosomes would arrange. And then you see what are the results in metaphase of meiosis 2, and then the various combinations A, B, C, and D that are finally produced in the gametes. So independent assortment of chromosomes in human beings. In human beings, because we have the haploid number of chromosomes is 23, n equals 23. So what are the different types of possible combinations? There are about 2 to the power of 23, or about 8 million possible chromosome combinations. That means a single man and a single woman can produce zygotes with 64 trillion combinations of chromosomes.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Crossing over is another event that produces genetic variation and it occurs during prophase 1 of meiosis. In this event, there is exchange of genetic material or chromosomal segment between non-sister chromatids of homologous chromosomes. So because of crossing over, gametes arise with chromosomes that are partially from the mom and partially from the dad. And these chromosomes are called recombinant because they result from genetic recombination. This figure illustrates the results of crossing over during meiosis for a single pair of homologous chromosomes.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
An important question is, do all animals reproduce sexually? Although some animal species can reproduce asexually, but very few animals can actually reproduce exclusively by asexual reproduction. So to find an answer to this, researchers studied a group of animals which are known as Deloid rotifers. And for hundreds of years, there is no evidence of males or any type of sexual reproduction in these animals. So scientists compared the sequence of segment of genes in Deloid rotifers and in closely related family of rotifers who are known to be sexually reproducing organisms. So in most species, what happens is the 2 versions of a gene that are present on a pair of homologous chromosomes, they're very similar because of exchange of genes during sexual reproduction. Now, if a species has survived without sex for millions of years, then what happens is that the DNA changes that are happening in the genes on the homologous chromosomes, they should accumulate independently. And the 2 versions of the genes would be very different from each other. They would get more and more different over time. This figure illustrates an experiment to determine whether Deloid rotifer chromosomes behave like those of sexually reproducing rotifers. The results were striking. The sexually reproducing rotifers, the homologous versions of the genes were very, very identical, 99.5%. So the difference was only 0.5%. On the other hand, the versions of the same gene in deloid rotifers were only about 46 to 96% identical. That means a difference of 3.5 to 54%. So these data strongly supported the evidence that deloid rotifers have evolved for millions of years without any sexual reproduction. What happens if something goes wrong in meiosis? Well, this can change the number of chromosomes, right? And when chromosome number is changed, that means the amount of genes are changed, and these are all genetic abnormalities. And these genetic abnormalities could be mild, or it could even cause death. It could be fatal. So when the members of a homologous chromosome, they fail to separate, that process is known as non-disjunction. So in anaphase, the two homologous chromosomes should separate, but when they don't, then it is known as non-disjunction, and it produces gametes with abnormal number of chromosomes. Now, non-disjunction could occur at meiosis 1 or meiosis 2. And this is involved in 10 to 30% of human conceptions and is the main reason for spontaneous abortion or pregnancy loss. There are two types of non-disjunction. Non-disjunction can occur in meiosis 1 or in meiosis 2. If it happens in meiosis 1, this is because the homologous chromosomes fail to separate, while if it happens in meiosis 2, this is because the sister chromatids fail to separate. And the figure shows what are the different types of abnormal and normal gametes that could arise due to non-disjunction. This figure shows fertilization after non-disjunction event in the mother. Down syndrome is an abnormality which is caused by non-disjunction in meiosis. This is genetically known as trisomy 21, which means that the individual who has Down syndrome has 3 number 21 chromosomes, making total of 47 chromosomes in that individual cell. So when there is trisomy 21, the disease is Down syndrome, which is the most common abnormality, chromosomal numerical abnormality, as well as the most common serious birth defect in the US. The figure shows the trisomy 21 in the karyotype. Symptoms of Down syndrome are varying degrees of developmental delays, shorter lifespan, and characteristic facial features. Although it is not known why, the risk of Down syndrome increases with the age of the mother. So the fetuses of pregnant women aged 35 and older are candidates for chromosomal prenatal screenings. Non-disjunction in meiosis can also cause abnormality in the sex chromosome numbers X and Y. And it is seen by researchers that whenever there is an abnormality in the sex chromosomal numbers, it seems to upset the genetic balance less than the unusual numbers of autosomes. The table here gives the names of some abnormalities of sex chromosomes. For example, if it is an XXY, the syndrome is called Klinefelter syndrome. XO, it's Turner syndrome. In case of human beings, as long as there is a single Y chromosome, then irrespective of how many X chromosomes that individual might have, that individual is a male. So this slide gives you the advantages of asexual and sexual reproduction. So first, asexual reproduction, this just totally eliminates or gets rid of the need to spend energy to form the gametes and finding a sexual partner. It does have an evolutionary advantage when organisms are suited to a stable environment. They don't need the genetic variation. Whereas sexual reproduction can offer an evolutionary advantage by producing offsprings that have a variety of genetic makeup and reducing the incidence of harmful genes more rapidly.